today's lesson, we will be looking at the chapter Cell Nuclear and Division. The learning outcomes for today's lesson would be: the students are to explain the importance of mitosis in growth, repair, and asexual reproduction. Furthering, students are also to explain the need for production of the genetically identical cells and fine control of replication. And the last learning outcome, the students are supposed to explain the meanings of the terms haploid and diploid. Also explain the need for a reduction division prior to fertilization in sexual reproduction. Shall we begin? Now class, as you all have learned in your lower secondary, the mitosis is an important process. Today, we will be focusing what is the importance of mitosis. First importance of mitosis would be the growth. When two haploid cells fuse together, they form a diploid cell. And if the new organisms to resemble its parents, all the cells that grows from this original cell must contain the same genetic information, and this can be only done with mitosis. But for your information, in mitosis, only certain part is expressed in any one cell. For example, epithelial cells, phloem, xylem, and these cells have to divide by mitosis to perform their particular function. This part of the lesson shows to you that the mitosis process will only express a general cell replica of their parent cell. But for the particular function of each cell, the cells have to go through further mitosis. The second function of mitosis is the repair, whereby exact copies of cells will be produced to replace the cells that die or damaged. And lastly, asexual reproduction, which is also a significance in mitosis, because they produce offsprings that are identical to their parents and to each other, and in this case we assume that they have no mutation. Now, in asexual reproduction, it has one main advantage. The parent cells are usually well suited to the environment and conditions where they live, therefore the offsprings can also thrive in this condition unless there are changes. And asexual reproduction would be a relatively rapid form of reproduction where large members can be quickly built up and the local area colonized. Asexual reproduction more common in plants. Asexual reproduction in plants, examples: tuber in potato, rhizomes in ginger, roots in banana, leaf in bryophyllum, and more other examples. They are also important in the cloning processes, especially in the tissue culture of the plants. Next, we are seeing what is the meaning of mitosis in maintaining a genetic stability. We saw in beginning that the daughter cells produced in mitosis process are genetically identical to the parent cell. So that can be achieved because the mitosis involves duplication of genetic material and it's equally distributed to each of the daughter cells. Therefore, the genetic stability is maintained as the number of chromosomes per cell is conserved, the replication of the DNA results in two identical chromatids, and the quality is conserved. The replication of DNA occurs before the cell divides. This is happening in the S phase of the interphase. And this is mainly to conserve the quantity of genetic material per cell. The terms diploid shows the cells contains two sets of chromosomes, as in there are two chromosomes that determine the same characteristics, one from the father and one from the mother, and the symbol of diploid is 2N. In mitosis process, the diploid 2N cell will go through mitosis and produce another diploid cell which is identical to the parent cell. Next, the haploid. The haploid term is defined as when the diploid cells divide by meiosis, it produces 4 nuclei, each with half the number of chromosomes, symbolized as N. The haploid condition can be seen in most organisms in their gametes, where they are always haploid. In humans, the gametes are the sperm and the ovum, both for the humans and the plants. So the diploid condition cells of the parent will go through meiosis to form a haploid cell. This basically differentiates the diploid and the haploid condition. Here, it shows a comparison of the life cycle in most animals and most plants. Let's see the life cycle of most animals. The life cycle of most animals show the diploid conditions to have a mitosis process, where from the zygote they go to repeated mitosis to form an adult. And in the adult, in the gametogenesis, the gametes are produced via meiosis. That has been produced are the sperm and the ovum, which is in the haploid condition. The fusion of the sperm and ovum would produce the zygote. In the life cycle of the plants, the diploid conditions see both mitosis and meiosis. But the difference would be they would have an extra life stage called as the sporophyte, where the sporophyte will go through meiosis to form a spore, unlike the animal cell which has an adult in the diploid condition. Then the spore goes through a mitosis stage to form a gametophyte. Gametophyte stage then will go through mitosis to form the male and the female gametes, and by the fusion of them they form the zygote. Now that you have understood all the things that I have taught you, let's do a small exercise. The stage when the cell is not dividing is called as the... That's very good. The answer is interphase. Let's see. The first stage of mitosis is called the prophase. During this stage, in animal cells, two cylindrical structures called the... You got the correct answer. The centrioles will move to the opposite ends, also known as the poles of the cell. Thin threads called the microtubules develop which span the cell from end to end and together form the... Very good. Spindle fibers. Towards the end of this stage, you must think something, because now, after the prophase phase, the chromosomes has to... the end of the prophase, nuclear membrane and the nuclear envelope has to break down. Once the nuclear envelope breaks down, the nucleolus disappears. Let's see the second part. During the second stage, called the metaphase, the chromosomes arrange across in the center of the cell, which is also called the metaphase plate or the equator. By the third stage, called the anaphase, The centromere of each chromosome divides into two, and the microtubules attached to each pull the individual chromatids or daughter chromosomes to opposite ends of the cell. In the final stage, known as the telophase, the daughter chromosomes disappear, a nucleolus reforms, and the cell divides into two. Thank you for joining the lesson. Hope we will see you soon. Take care. Bye.